Hello students, welcome to UGC's EPG Pathshala. Myself Dr. Pranita Roy, an assistant professor at Amity Institute of Biotechnology under Amity University, Noida. I am here to discuss on the topic applications of genetic engineering. Genetic engineering has broad applications, but before highlighting the various applications, let us first understand what is genetic engineering. Genetic engineering broadly means the manipulation of an organism's genome to make useful products. It is a set of technologies used to change the genetic makeup of cells, including transfer of genes within and across species boundaries to produce improved or novel organisms. So in this module, we are going to learn the concept of genetic engineering, the different approaches and strategies of genetic engineering, the various applications of genetic engineering, and then understand the underlying ethical considerations related to genetic engineering. So now I am going to discuss the concept map of genetic engineering. Genetic engineering is basically a collection of techniques that alter the genetic constitution of cells or individuals. It mainly requires 4 basic steps of RDT or molecular cloning. The 4 steps includes isolation of DNA fragments, joining the DNA fragments to a suitable vector, introduction of the vector into a host cell, and finally the selection of colonies with the desired sequence. And this leads to the generation of genetically modified organisms. These genetically modified organisms, they find application in basic research, food industry, pharmaceutical and health, environmental restoration, crop and animal trait improvement. But all these advancement has also got some drawbacks which raise some legal and ethical questions. There are basically 4 stages to any cloning experiment. These are the generation of DNA fragments, joining the fragments to a suitable vector to produce recombinant DNA, propagation of the recombinants in a host cell, and the identification and selection of the required sequence based on the presence of the foreign genes getting expressed in the host. Now I am going to discuss the general strategy for gene cloning. To achieve the aims of genetic engineering, a cloning strategy has to be devised that will enable efficient use of the technology. There are basically 4 stages to any cloning experiments. These are the isolation and preparation of DNA fragments, production of recombinant by joining the fragments to a suitable vector, introduction of the vector into a host cell, and finally the screening of the recombinant host carrying the desired gene. The successful completion of gene cloning results in a specific DNA sequence which may be commercially applied for a variety of purposes like production of recombinant proteins, genetically modified microorganisms, transgenic plants and transgenic animals. The most suitable bacterial host is Escherichia coli because of the complete knowledge of their gene structure, the faster growth rate, easy to culture and handle. Besides the commendable characters of E. coli as a suitable host, the isolation and purification of the product becomes difficult owing to the presence of inclusion bodies in the host cell. Also, the expression of a eukaryotic gene becomes problematic in a prokaryotic host as many key elements of eukaryotic gene expression are generally absent in prokaryotes. These are mainly the chromatin and small RNase regulation, the pre-messenger RNA processing, the lack of intron region, the lack of RNA splicing machinery, and post-translation modification system. By cloning a cDNA form of the eukaryotic gene, the problem of intron can be avoided and incompatibility problems can be avoided by using eukaryotic cells as hosts. The use of eukaryotic host is the preferred choice, and for this reason yeast, insect and mammalian cells are encouraged. The use of eukaryotic host is the preferred choice, and for this reason yeast, insect and mammalian cells are encouraged. Yeast cells are considered to be a suitable host as they are being single cell fungi, they are easy to grow unlike most eukaryotes. The genome of yeast is designed to form yeast artificial chromosomes by combining the essential component of a eukaryotic chromosome like an origin site of replication, a centromere and 2 telomeres which can be ligated with foreign DNA and thus serve as a suitable vector. However, eukaryotic proteins which require specific modifications, the successful cloning requires to be carried out in animal host. Dealing with animal and plant host, the problem lies in the introduction of the foreign DNA into the organism. Hence, different techniques are employed to facilitate entry of foreign DNA into eukaryotic cells. Some of these techniques involve direct DNA uptake, electroporation, microinjection, and entry by biological means, which involves the use of infectious agents such as lentiviruses to transfer genes to animal cells or agrobacteria to transfer genetic material to plants. Once the DNA finds its entry inside the cell, it is incorporated into the cell's genomic DNA by genetic recombination. Genetic engineering has led to a number of successful GMOs. With advancement, it is now possible to manipulate, remove and add genes to a variety of different organisms to induce a range of different traits. Herbert Boyer and Stanley Cohen in 1973 created the first genetically modified bacteria, followed by a number of remarkable achievements in the field. Some of the major achievements of genetic engineering are: in 1974, GM mice was created. In 1976, commercialization of the technology, after which producing and selling genetically modified foods and medicines began, which lead to a number of further GMOs. In 2003, began to sell GMOs as pets, which are named as glowfish. A number of notable achievements since then involves development of drugs such as treatments for cancer, development of transgenic insect resistant crops, and development of transgenic animals for production of growth hormones and pharmaceutical products. The expression of transgenes is possible in transgenic organisms as the genetic sequence for proteins are similar. The transgenic organisms are now developed to produce various substances such as foods, pharmaceuticals, biochemical, etc. Now it is possible to clone plants, fish, and even livestock. Now we are going to discuss the various applications of genetic engineering. The number of applications for genetic engineering are increasing as more and more is learned about the genomes of different organisms. Genetic engineering has various applications in food industry. Genetic engineering finds application in food industry, which is a result of modification of the genetic material of plants or animals. Many genetically modified whole foods or ingredients present in them available today are a result of gene modification. A number of enzymes are involved in fermentation and digestion of foods. This has led to the concept of production of recombinant enzymes from genetically modified microbes such as chymosin and lipase for cheese production and alpha amylase for flavor enhancement in beer industry. One of the latest technologies involves production of cow milk containing increased amount of a cheese making protein, casein, and foods without beta lactoglobulin by RNA interference technology. Genetically modified foods are obtained from genetically modified organisms or transgenic crops. Genetic engineering has resulted in a number of improved traits in transgenic plants by genetic alteration. Some of these traits are production of extra nutrients in the food, increased growth rate, disease resistance and herbicide resistance, better taste, increased shelf life, lesser requirement for water, etc. This was the first commercial genetically modified food. Genetic engineering mainly focuses on cash crops, a list of which is being shown here. Like rapeseed, it has been made pesticide resistant, cotton again pesticides resistant. A number of vitamin production is being done by such type of transgenic crops. Genetic engineering has also got wide application in pharmaceutical industry and medicine. By genetic engineering, a variety of medical products are available today. Among these products, insulin and human growth hormone were first commercially available products obtained from recombinant E. coli. Recombinant insulin is the result of successful genetic engineering. The initial production of insulin involved the separate synthesis of insulin A and B chains in two bacterial strains. Both the insulin A and B chain genes were placed under the control of lac promoter for inducible expression by lactose inducer. After purification of the A and B chains from the bacteria, the chains were then linked chemically to produce the final insulin. The production process of insulin is being shown here. Recombinant insulin is now commercially available in several forms and is involved in diabetes therapy. Now, I am going to discuss the general strategy for gene cloning. To achieve the aims of genetic engineering, a cloning strategy has to be devised that will enable efficient use of the technology. There are basically 4 stages to any cloning experiment. These are the isolation and preparation of DNA fragments, production of recombinant by joining the fragments to a suitable vector, introduction of the vector into a host cell, and then finally the screening of the recombinant host carrying the desired gene. The successful completion of gene cloning results in a specific DNA sequence which may be commercially applied for a variety of purposes like production of recombinant proteins, genetically modified microorganisms, transgenic plants and transgenic animals. Genetic engineering techniques have been applied in numerous fields including research, agriculture, industrial biotechnology, environment and medicine. The number of applications for genetic engineering are increasing as more and more is learned about the genomes of different organisms. A few interesting or notable application areas is being discussed here. The production of pharmaceutical products from transgenic animal is considered as farming. Farming involves the use of genetic engineering technique to insert genes into host animals or plants resulting in expression of useful pharmaceutical products. The development of transgenic super mice in 1982 led to the wide application of farming in pharmaceutical industry. Human drug tissue plasminogen activator, a valuable therapeutic protein to treat blood clots, was first produced from super mice as a result of genetic alteration in 1987. Since then, farming has resulted in a plethora of recombinant products made in E. coli, a list of which is being shown here. The recombinant proteins produced by farming act as drugs for various human diseases. These therapeutic products can be directly injected into the bodies of the patient to treat the disease and deficiency. The recombinant vaccines are an important group of therapeutic products. A number of vaccines are now available for animals and human, which is going to have a major impact in the healthcare industry. One of the initial vaccines produced by recombinant DNA technology involves the cloning of the surface antigen of the hepatitis B virus in the yeast Saccharomyces cerevisiae under the control of alcohol dehydrogenase promoter. A number of recombinant vaccines are now commercially prepared by the recombinant DNA technology, where only the outside coat protein of the microorganism is expressed in the host to create the vaccine. The expressed protein can then be purified from the recombinant host and used for inoculation. This method has the advantage of safe delivery of antigen without transferring the actual disease causing microbe to the host. Currently, recombinant vaccines for the hepatitis B virus, herpes type 2 virus and malaria is under trial for use in future. The latest development involves the production of edible vaccine using transgenic plants as a delivery mechanism, which involves the presence of vaccine in the edible part of the plant. This technology has tremendous potential as it enables easy delivery of vaccine by just consumption of the edible part. The trials for development of a vaccine containing banana or tomato are currently underway. With the advancement of genetic engineering, it is now possible to treat the genetic defects by the replacement of the defective gene with a functional copy by gene therapy. This technique has great potential in the treatment of genetic diseases. The gene therapy protocol can be made effective by the following approaches. These approaches involve the insertion of a normal gene to compensate for a non-functional gene, repair of an abnormal gene by selective reverse mutation, alteration in the regulation of gene pairs. Gene therapy involves the following steps. Delivery of the therapeutic gene by a suitable vector into a target cell of the patient, infection of the target cells with the viral vector, insertion of the genetic material into the target cell by recombination between the defective gene and the functional copy of the gene, and finally expression of the functional proteins from the inserted gene resulting in a normal cell. A number of genetic disorders caused by single gene defects such as cystic fibrosis, muscular dystrophy, hemophilia, sickle cell anemia, and AIDS can be treated by gene therapy approach for which the clinical trial is underway. However, there are some problems associated to a successful gene therapy. These are introduction of large segment of DNA to the right side on the genome become problematic. Sometimes the defective mechanism of the host destroys the genetically altered cells. Genetic engineering has thus resulted in different kinds of vaccines, antibodies and vitamins, drugs and hormones which are easily available in the market and are involved in the treatment of many diseases. Genetic engineering has wide application in environmental biotechnology. It is exploiting the huge potential of microorganisms, plants, animals for the restoration of the environment. It is actively involved in the development of microorganisms and biocatalysts for remediation of contaminated environments and in development of eco-friendly processes such as developing recombinant strain for biofuel production, etc. Some of the areas where genetic engineering is involved is being shown here. A number of genetically engineered microorganisms are developed which are involved in the biodegradation of waste material. As the genes for enzymes involved in the biodegradation pathway are mainly located on the plasmids, it is possible to create new strains by genetic manipulation of such plasmids. Using this technique, Chakrabarti and his team of co-workers in 1970s developed a new strain of bacterium, Pseudomonas, named as superbug. This superbug is able to produce a combination of enzymes involved in degradation of a number of hydrocarbons present in petroleum. Superbug became the first genetically engineered microorganism to be patented. Keeping this in mind, new recombinant strains of Pseudomonas have been developed which transforms a number of toxic chemicals such as hydrocarbons, chlorinated solvents, polychlorophenyls and metals in a less toxic form. There has been a series of development of recombinant microorganisms specially designed for the degradation of environmental pollutants. A summarized list of such microorganisms is being shown here. The problem of soil pollution caused due to increased use of herbicides, pesticides and insecticides can also be solved by using recombinant microorganisms. The overuse of chemical herbicides, pesticides and fertilizers are detrimental to the environment. So, attempt is being made to develop bacterial and viral pesticides which will help in reduce use of chemical pesticides. Genetically engineered bacteria in which toxic genes from Bacillus thuringiensis is cloned are used as biological pesticides. Genetic engineering also finds application in crop improvement through transgenesis. Crop plants have been the focus of genetic engineering as efforts are being made to improve the traits of plants. Transgenic plants are developed for the following reasons. These are: gene insertion may result in improvement in the agricultural or commercial value of a plant. Transgenic plants can act as a living bioreactors, facilitating production of commercially important proteins or metabolites. Transgenic plant helps in the understanding the function of different genes. A number of genes can be combined with crops to produce desirable properties such as herbicide drought resistance, higher yield, tolerance towards cold, drought, salt, etc., faster growth, improved nutrition, delay of senescence, longer shelf life, increased post-harvest shelf life, altered flower pigmentation, nitrogen fixation capacity to fix atmospheric nitrogen. The creation of a transgenic plant involves the TI plasmid isolated from tumor-inducing bacteria is used to transfer the gene. The modified plasmid is introduced into a cell, and finally the plasmid recombines with the plant genomic DNA. One remarkable development in transgenesis is the development of resistance against insect by introduction of a gene responsible for the protein protoxin from Bacillus thuringiensis. Also, transgenic plants can be designed to produce a variety of useful compounds like therapeutic products and metabolites. Recently, transgenic crops with combined traits like herbicide tolerant and insect resistant has been developed. Genetically modified plant products in pipeline are increased level of irons and vitamin A in rice, fast ripening process in banana, improved feed value in maize, high levels of flavonols in tomatoes, drought tolerance in maize. Genetic engineering has also led to trait improvement of animals through transgenesis. Genetic engineering involves the introduction of transgene into animal to improve the trait of transgenic animals. Transgenic animals thus finally express the trait of the introduced gene. Transgenic animals are also created to study the function of different genes to develop proper treatment of a disease. The mechanism of development of a transgenic animal includes removal of healthy egg cells from a female host animal and fertilization in the lab, the identification and isolation of the desired gene obtained from another species. The desired genes are injected directly into the eggs and then implanted in the host female. Transgenic animals can be a successful mean to provide an economical production of enzymes, proteins, quality and quantity improvement of meat and other animal products. With the successful cloning of Dolly, a sheep in 1997 by genetic engineering, there is a continuous effort in the direction of cloning of useful livestock. Some of the remarkable products developed through transgenesis in animals include production of human proteins in animal milk, production of biosteel, a high-strength silk product from goat's milk, growth of tissues on a 3D printer by a genetic manipulation of stem cells can be used as a skin substitute for wound healing, production of therapeutic proteins such as monoclonal antibodies from the milk of transgenic cows, goats and mice, which is used to administer drugs in various diseases. Genetic engineering and transgenic thus holds tremendous potential in the field of basic research and also commercial and industrial consideration of different products. The characteristics of genetic engineering possess vast promise but also involve some potential threat to mankind, bringing challenges to our ethical system and religious beliefs. The main reason genetically modified organisms are not more widely used is due to ethical concerns, which are genetically modified foods are not independently tested before the food is approved, so the health of the human is threatened because the consumption of it could lead to some allergic reaction. Another safety consideration is the health of farmers and their families, animals and communities who are put at risk with exposure to chemicals used in tandem with GMO seeds. The genetically engineered organisms which are released into the environment can be a threat to it because they can interact with other living organisms in the environment in an unpredictable manner. Genetic engineering can result in creation of toxic vegetation, posing serious threat to wildlife and may result in new strains of molds and fungi. It could also used to create biological weapons. The creation of pathogen resistance in plant may result in development of strong resistant variety of pathogens. There is a serious concern that animals will suffer as a result of being genetically modified, as use of growth hormones may cause limb deformation and arthritis as animals grow. Genetic engineering applied to human may have serious implications like parents opting for designer baby, that is deciding their children's eyes, colors, heights or even genders before birth. Another concern is that if tests are carried out for genetic diseases on unborn babies could lead to abortion if a disease is shown to be present. So, in this lecture we learnt about the description of genetic engineering, the different strategies involved in creation of genetically modified organisms and transgenics, application of genetic engineering along with the future development, ethical considerations involved with GMOs and transgenics. So, we have understood that the number of applications of genetic engineering are increasing as more and more is learned about the genomes of different organisms. However, the main reason genetically modified organisms are not widely used is due to underlying ethical concerns. So, research and applications of genetic engineering should proceed with caution and humility. Genetic engineering has proven that it is not a threat to mankind, but rather a powerful tool which holds tremendous promise for human well-being. Therefore, it is an understatement to say that genetic engineering will revolutionize the medicine and agriculture in future. Thank you. All the information related to my lecture is available at UGC ePathshala website.